side chain that's curvy got to do with scurvy? Proline. So the amino acid or protein letter proline is probably the weirdest of them all. So most amino acids, they're kind of just like charm bracelets on for a protein. And so they have this unique part that sticks off. And most of those unique parts just like kind of stick off. But prolines actually loops back and attacks the backbone again, so you get this kind of circle. What happens is that this is going to restrict the motion of regions of a protein that contain proline because you can't rotate around that backbone as much, and you're restricted to very particular angles, as we'll see. This can stratify regions of a protein, and so this is useful for, say, the COVID-19 vaccines, where it's used to stabilize the spike protein, so the coronavirus surface protein, in a conformation or shape that makes it so that our immune system can make really nice antibodies against it. In our bodies, a modified form of proline called hydroxyproline is actually a key component of the skin protein collagen. And so collagen, this hydroxyproline, forms this really cool, helps the collagen form this really cool like triple helix. And this triple helix is going to be really important for stratifying our skin, but in order to make it, you need to make hydroxyproline, and to make the hydroxyproline, you need vitamin C, and this is where you get the connection to scurvy, where if you don't have vitamin C, you can't make this modified proline, and so you can't make this strong skin. So those are just a couple cool things about proline, but so let's talk about more about how it can't make the normal types of structures that we think about, these alpha helices and these pleated sheets, because when you have that curving around to attack the backbone, you're kind of stealing the hydrogen that would normally be used to make hydrogen bonds that are responsible for making those structures. And also, if that wasn't awkward enough, proline can also do this thing called make a cis-proline bond, where basically the backbone actually gets rotated, and so you have it where instead of a zigzag, you kind of have a curve. We call this a cis bond, and it's really awkward, but it can actually be found, and I'll talk a little bit more about how this can be found in crystallography, and how sometimes it was like overfound when it wasn't really there, but crystallographers were like, well, that density looks weird, let's just call it a cis-pro. And so I'll talk a little bit more about Jane Richardson and how she taught me about cis-prolines. So that's the basics of what we're going to talk about, so let's dive in.
So this is proline. Like all of the other amino acids, it has an amino group and a carboxyl group, and it can in a unique side chain. And so the amino group and the carboxyl group, these are the generic parts that the amino acids use to link together to form chains that fold up into functional proteins. And the shape that those proteins take, and therefore what functions they have, is going to depend on the sequence of amino acids, because each of these amino acids also has a unique side chain or R group, this unique part that sticks off, kind of like a charm in a charm bracelet. And this is where with proline, we have that really, really awkward looking charm. There's a common, as I said, proline has an amino group and a carboxyl group. There's a common misconception that proline is an amino acid as opposed to an amino acid. So what do I mean? Well, if you had an imine, this nitrogen would be double bonded to a carbon. That's not what we have. Even in the case of proline, where that nitrogen is weird looking, it's got 2 bonds, 2 carbons, those are single bonds. So this is still an amino acid, but it's a different kind of amino acid. All of these other ones, we had a primary amine group, where here we have a secondary amine. And so what do I mean by this? Basically, we can classify a nitrogen, we can classify an amine based on how many carbon groups it's connected to. And so if we had a primary, we'd be only connected to one other carbon. And this is what we see in the case of other free amino acids, that they have this nitrogen that's attached to a single carbon, and then either 2 or 3 nitrogens, depending on the protonation state, which depends on the pH. So in the body, it's typically going to be in this protonated state, so where you have these 3 hydrogens. But in the case of proline, what you have is a secondary amino group, because you're attached to 2 carbons, and therefore its protonated form, it only has 2 hydrogens. So why am I harping on about this? Well, it matters. And it's not just a terminology thing. It matters because you're going to have one less hydrogen there. And when we talk about the amino acids linking together, when they do so, that hydrogen is going to be really important, the hydrogen that's normally there. And in the case of these amino acids, when you link together primary amino acids, what you're left with is you still have a hydrogen group sticking off. And when you have this hydrogen group sticking off, well, this is going to allow it to form these things called hydrogen bonds, which are going to be crucial for giving the protein its secondary structure. So we're talking things like beta strands, which can team up into beta pleated sheets, as well as alpha helices. All of these are going to depend on having a hydrogen on that nitrogen. When you had a primary amino group, when those link together, you're going to it's going to be deprotonated. So you're not going to have a charge on this nitrogen anymore, because instead what's going to happen is it's going to, in this amide bond, it's going to form this really stable bond. And in order to do this, it's typically going to be deprotonated. And so then you have the lone pair of electrons that can then be participating in this resonance. So above the scope of the post. But just know that although it's protonate, you'll see these end amino groups protonated. In the backbone, you're not typically going to see these protonated, or you're not going to see them like double protonated. So you'll have one hydrogen, but you're not going to have two. You're not going to have that charge, because instead you're using those electrons to participate in this resonance. And we'll get much more into what I'm talking about here. I just don't want you to get confused how we're going from having 3 protons to having 2 protons. But here, just know that for the backbone, you're typically going to have 1 hydrogen sticking off. In the case of proline, however, because you started with 1 fewer hydrogen, well, now you're not going to have that hydrogen sticking off of the backbone. And if you don't have that hydrogen sticking off of the backbone, well, now you can't participate in that hydrogen bonding, which requires, well, a hydrogen. And so what am I talking about with this hydrogen bonding? So we're going to get kind of technical into details about atoms and bonds and interactions, but just to get the gist, the key things to keep in mind, and even if you want to know the details, the core things to remember to help you really get a fundamental understanding of it are simple things. So basically, opposite charges attract and like charges repel. And this is true for full charges, but it's also true for partial charges as well. And so even in the case where we don't have any full charges, so if we're just looking at this, we don't see any full charges. But what you're not seeing, what is there, but that you're not seeing in this diagram, is that there's actually partial charges. And there's partial charges because you have uneven sharing of electrons. And so basically, all of these different circles, these carbons and these hydrogens and these nitrogens, these are representing atoms, which are the basic units of elements. And these are made up of smaller parts, and those smaller parts have charges. So even if an atom is neutral overall, it still has those charged parts inside of it. They just cancel each other out. And so these charged parts are going to be protons, which are positively charged, and electrons, which are negatively charged. There's also neutral things called neutrons, which are going to kind of help glue together the protons in a dense central core called the nucleus. And then you're going to have electrons whizzing around them. What happens is that each of these elements has like a number of electrons that they want. They want to have like this full outer shell, which I'm not really going to talk about here, other than to know that there's a certain number of electrons that they want, and they can kind of swap with each other, or they can share with each other in order to get what they want. When we talk about sharing with one another, this is how we form covalent bonds. So atoms can kind of say, okay, you want one atom, you want one electron, I want one electron. We both have one electron. Let's kind of like share our electrons. And so what they do is they kind of merge their electron clouds. So remember, these electrons are kind of just like whizzing around in this cloud, and they can kind of overlap their clouds in a way that allows them to share those electrons and therefore form bonds. Now, they're sharing, but they might not be sharing fairly. And this depends on how electronegative the atoms are. So electronegativity is kind of like how strong of a pull they have on those electrons. So if you think of those electrons as kind of like dogs that these atoms are walking, and the actual like leash is being held by the protons. So the protons are those positively charged particles and the electrons are those negatively charged particles. And going back to our opposite charges attract, well, the protons are going to be attracting those electrons and kind of holding them in. Different atoms are going to have different numbers of protons, and they're also going to have different like distances that those electrons are from the protons. And so because of this, they're going to have different amounts of pulling power on those electrons. And so we call this pulling power electronegativity. As you move to the right across the periodic table, you're getting more electronegative because you're gaining more protons. So the number of protons defines an element. So say nitrogen has 7 protons and oxygen has 8. And that's what those numbers are up there. And so the more protons you have, the more pulling power you have. And so if you look at a nitrogen and you look at an oxygen, these are going to have more pulling power than a carbon. Similarly, the further away the conversely, the further away the electrons are, the less the pull is going to be. So if you have something, when we look back at here, we'll see that when we get to bigger elements, these electrons are going to be further away, so there's going to be less pulling power. But when we're dealing with carbon, nitrogen, and oxygen, these are going to be in the same row. And so we can then just say that oxygen is going to be more electronegative than nitrogen, which is going to be more electronegative than carbon. And this is going to be because they have more protons. In the case of carbon and hydrogen, they share pretty fairly. So but hydrogen only has like a single proton, so it doesn't have that much pulling power, but that proton is really close to the electron. And so when we say that we have a carbon bound to a hydrogen, we typically call this a nonpolar bond. And so polarity is when you have a separation of charge, and you get that separation of charge if you have uneven electron sharing. Because hydrogen and carbon have this fair electron sharing, you're not going to get polarity. But if you have a carbon or if you have a hydrogen bound to a nitrogen or bound to an oxygen, well, now you're going to get polarity because oxygen and nitrogen are electronegative. They're going to pull those shared electrons away. And when they pull those shared electrons away, you're going to get a partial negative charge where those electrons are being pulled to, and a partial positive charge where those electrons are being drawn from, because those protons are still there and those protons are still stuck in place. So we get the separation of charge we call polarity. And remember, opposite charges attract. What's going to happen now is that you can have these attractions between a hydrogen that's bound to something electronegative like an oxygen or a nitrogen, and an oxygen or a nitrogen, which is going to be partly negatively charged. If we have this special case of this, where we have a special case of this, we call a hydrogen bond or an H bond. Now we can get those like dipole-dipole interactions, so the dipole with the polarity, the partial charge regions. We can get those type of interactions even if we don't have a nitrogen, if we don't have a hydrogen in this distinct conformation, in this distinct type that we call them hydrogen bonds. But in a hydrogen bond, it's like a especially strong type of those dipole-dipole interactions. And it involves when you have a lone pair of electrons on an oxygen or a nitrogen. So this is going to be an unpaired electron. So remember, those electrons, they can share, they can link up through sharing electrons to form these bonds, but they can also just have like lone pairs. So where they just have electrons that aren't being shared, but they're just owned by themselves. And these can also participate in hydrogen bonding. And so in the case of an oxygen or a nitrogen, you often have these lone pairs of electrons, which are then going to be able to participate as what we call a hydrogen bond acceptor. And so they're accepting the hydrogen bond from this hydrogen, and the hydrogen is going to be attached to an oxygen or a nitrogen or something electronegative, and this is making that hydrogen partly positive, and this oxygen has got those 2 electrons is going to be partly negative. And therefore, if you have them in this distinct orientation where they can really line up their clouds nicely, even though their clouds aren't really like overlapping like you would see in a covalent bond, you're still going to get a strong interaction, stronger than the typical like just interactions that you would normally see. And we call these hydrogen bonds or H bonds, and we see them all over the place in biochemistry. A common place that we see them is in like the base pairing between different strands of the letters in different strands of DNA and RNA. And we see them in the peptide backbone. And this is how we get our alpha helices and our beta strands and sheets. Why this is that in your peptide backbone, remember we have all of those like similar units that are linking up with those same generic backbone. And in that generic backbone, except in the case of proline, we have it where we have a hydrogen and an oxygen. We have a hydrogen on a nitrogen, and then we have this oxygen with these lone pairs, and we have them in this regularly repeating pattern where we'll have an acceptor, and then we'll have a donor, then we'll have a carbon, and then we'll have an acceptor and a donor and a carbon, and an acceptor and a donor and a carbon. And because it's laid out in this distinct repeating manner, you're going to have characteristic structures that arise when those when the chains, the peptide chains or the regions of those chains are kind of aligned. They can do so in a way that allows them to form these like stereotypical interactions that are then going to give this protein its structure, and these interactions are going to make it so that the structure is really strong in the region where you have all those hydrogen bonding, kind of like gluing these strands together. And remember, because you have that orderly array where you have that distinct repeating pattern, you're going to get these distinct shapes. However, this is going to be limited by the nature of the side chains in some cases. So the rotation around the peptide backbone is going to be limited to basically rotating around a series of planes, because you can't rotate around the peptide bond itself. And so much more on this in other posts, but you can't rotate around this amide bond, this peptide bond, because it's going to have this, this partial double bond character. It's going to have this electron delocalization or resonance, where basically instead of just sharing between the neighboring atoms, all three of these electrons are going to be sharing, or all three of these atoms are going to be sharing electrons. And in order to do so, they have to lie in a plane. And so you can't rotate around this bond, but you can rotate here or you can rotate here. And this is going to help limit the rotation and also contribute to this secondary structure. But the secondary structure and the rotation itself is going to be limited by the R group, so by the unique part of the side chain. And so we looked and we saw how with glycine, because it was really small, it was able to take on more angles than other amino acids. And in the case of proline, we're going to see that although it's not as big as it's one of the smaller amino acids, it's going to be the most restricted, because you're going to be, you have this awkward side chain that really has limited flexibility in its backbone. And we can represent this limited flexibility, or we can like display this limited flexibility in something called a Ramachandran plot. And it's going to basically show you the peptide backbone dihedral angle. So a dihedral angle is an angle between planes. Remember, with these peptides, we're going to have a chain of planes that are kind of like connected. And in the case of proline, these angles are going to be very limited, the angles you can rotate between those planes. And so if we then plot out the angles, we can see that we get this really weird looking plot. So proline is going to have very restricted movement. Not only is proline restricted in the rotation, but also remember, it doesn't have that hydrogen in its backbone when it links up. When it links up, it loses that hydrogen. And so when it loses that hydrogen, it's not going to be able to serve as a hydrogen bond donor. And so you're not going to be able to form these interactions that stabilize these beta strands, or they stabilize the helix. So it's going to mess up this whole like interconnected network. In addition to adding a sort of kink, it can't form a stabilizing interaction. And so you're not likely to find a proline in a sheet or in a helix, but you are more likely to find it in the connecting region. So in these like tight curves, tight turns between these regions. And sometimes you often see it like linked up with the glycine, which is really small. So you can kind of like help make it so that you have a better turn, and you don't have steric hindrance. So you don't have things like groups kind of like clashing into one another. And so you'll see proline kind of like in these these sharp turns often. Having a proline is able to, it's not just like a loosey-goosey loop though, remember? So it's like a sharp loop kind of, and it's a sturdy one. And so we can take advantage of proline if we want to like stabilize regions of a protein. And this was actually used in COVID-19 vaccines, as well as solving the initial structures of the COVID-19, the coronavirus's spike protein. And so the spike protein is the protein that sticks off on the surface of the virus, and it's basically going to allow it to dock onto cells. What happens is that it undergoes this shape change, and this shape change is going to kind of flip up a region of the protein, and then it's going to allow it to latch onto the cell membrane and fuse the membranes, and much more on this in other posts. But in order to undergo this shape shift, basically this region here, it kind of has to like unhinge. And what happens is that if you introduce a proline mutation, so if you switch one of the amino acids that is there to a proline, well, now you can lock it into a position where you can't unhinge it as easily. Now, why would you want to do this? Well, if you stabilize it in that state, that like pre-fusion conformation, then it makes it better so that your immune system can learn to make antibodies against that shape, that you then want the immune system to be able to recognize, so that you can make antibodies that are then going to block it from docking onto cells. And so, say for the Novavax vaccine, where it introduces the pre-made spike protein, they use a proline substitutions in order to make it so that it's stabilized in that pre-fusion state. And this was also crucial for solving the initial structures of the spike protein. So Jason McClellan's lab was able to get structures of the spike protein really, really quickly, because they knew from prior work with MERS that there were specific places that if you introduce proline mutations, you can stabilize it. And then later work, they would show that like if you introduce even more proline mutations in different places, you can get this like hexapro, which is even more stable. And so this was really crucial to getting those initial structures, because if you didn't have them, then the protein would be too unstable, and it would be flipping into that like post-fusion state where it's kind of like flipped up. So we have proline to thank for the science and the vaccines and all of that various stuff. When we were looking at these graphs, you might have been wondering, okay, well, where did these angles come from? How do we know this? These are typically taken from X-ray crystallography, and so much more in X-ray crystallography and other posts, but it's a core method of structural biology, where we use it to figure out the structures of proteins at the atomic level. So basically, what do they look like? So all the structures of the spike protein I was just showing you, well, a lot of those were from a lot of the structures of the spike protein are actually from cryo-EM, so cryo-electron microscopy, but some of them are from X-ray crystallography. And X-ray crystallography is still a core way that we do structural determination. So basically, we're trying to figure out where all of these atoms are. And what happens is that we kind of get the protein to arrange itself into these like crystals, where it's basically an orderly lattice. So these proteins are kind of like stacked. It's kind of like a brick wall with the proteins, where they're all in the same orientation. And then we shine X-rays at them. And these X-rays are these really energetic rays that are going to bounce off of the different electrons in the protein. And those waves are going to interact with one another. And when they interact with one another, sometimes they'll add together and sometimes they'll cancel out. But what you get is this pattern of spots in the detector. And we call this the diffraction pattern. And then we can work backwards from that spots in order to figure out where the electrons were and therefore where the atoms were. So what those X-rays are bouncing off of is the electrons. And so, but then the electrons are kind of like circling around the protons and the nuclei of the atoms. So then we kind of model in where the backbone of the protein was, and the side chains and the different atoms and stuff. But so it all works it's really nice if it all works out well. But what happens is that if you have you're not actually getting directly the shape of where the atoms are. You're getting where that electron density was. And depending on how good your structure is, you might get like a really weak signal, or you might get a really strong signal. And the signal you see, it kind of looks like this meshy thing. And so you can see that it can be kind of hard then when you try to model in, okay, how can we draw in the actual atomic positions in this mesh? If you have a really high resolution, basically it's easy to do this, but if you have a really low resolution, you have a kind of like blobbier mesh, it can be harder to do this. And so if you have a really high resolution structure, then you're able to figure out those angles really well. If you have a lower resolution structure, you might not even be able to tell which amino acid is in which place. Why this comes into play is this thing called cis-prolines, or well, why I wanted to bring this up is this thing called the cis-proline bonds. And this was a topic that I learned about from Jane Richardson, who was the inventor of the ribbon diagram, or the ribbon diagram notation or representation for drawing proteins, where we draw the helices as these cool helices and the beta strands as these like this. And she also developed this software with her husband called Molprobity, which helps point out potential problems in those structures. So when scientists are trying to build in the models, so trying to build in those sticky figures into that electron mesh, the software helps you check and make sure that it's like geometrically feasible. And one of the things that it checks for is this thing called a cis non-pro or even a cis-proline bond. And so what this involves is the orientation of the peptide bond, the backbone, like actually in that peptide bond, kind of. And so basically that backbone, it typically zigzags. So where you have the neighboring alpha carbons, so the alpha carbon is going to be the carbon that's attached to the unique part of the side chain. Now, normally in a peptide bond, what we have is a trans peptide bond, where those alpha carbons are going to be on opposite sides of the peptide bonds. Now, in cis, however, you would have it where they're on the same side. Now, normally this is unfavored because you're going to get steric clashing. So when steric, this is basically things can't occupy the same place in space. And so if you have steric hindrance, two things are going to be trying to kind of like occupy the same place and hindering one another. But typically, if you have a trans orientation, that's going to avoid having that hindrance as that too bad of a hindrance. But if you have cis, well, now this is going to be really uncomfy. And so normally proteins don't want to do this. Amino acids are going to be really unhappy like this. It turns out that proline can kind of tolerate this. Basically, it's going to have such a weird backbone already that making it cis isn't that much more uncomfy than making it trans. But it doesn't do this that often, because in order to actually form the cis, you need to actually have an enzyme help. So an enzyme is like a reaction helper or speed upper. Because what we're having here is basically when the ribosome adds the amino acid. So the ribosome is going to go along the messenger RNA instructions and piece together those amino acids to form the protein. When it's doing so, it's putting them in the transfiguration. And so in order to change it to the cis, you would have to have help. And you have to have help because this involves actually rotating around that peptide bond that we said you can't rotate around. Remember, that's the bond that's super stable. You have that resonance, you have that electron delocalization. And so this is like, this is in that plane that we said you can't rotate in the plane. And now we're saying, okay, well, you can rotate in the plane, but you can only rotate in the plane with the help of this enzyme, because the enzyme is going to allow you to like kind of break that bond, break that resonance and allow it to like flip over. And so you need this enzyme help, but there are cases where this does happen. And so this peptide prolyl isomerase is able to do this, and this can actually play important roles, this cis-proline. And so, but when signed, and it's often preceded by glycine or an aromatic amino acid. So this cis-proline does exist, and because it's not that much unfavorable than the trans, but it needs help to exist. And so it's not just going to exist all over the place. However, when scientists first discovered that this cis-proline was actually a thing, they got really excited, and then basically they started identifying it everywhere, even when it wasn't really there. So only about 5% of prolines are cis, and only about 0.03% of non-prolines are cis. What happened is once scientists kind of recognized that the cis-peptides could exist, when they went back and they had their structures, the structures of the amino acids, and they had, or the crystal structures and things, and they had these kind of like weird meshes that they had trouble fitting things into, they're kind of like, oh, well, if we make it cis, we can fit it in there. And so there were a lot of false cis peptide bonds identified in the early days. And then thanks in part to Jane Richardson and David Richardson, her husband, and their mold probity software, they were kind of able to get this problem under control. So now there's fewer false cis peptides being recognized. And let me just, before, in case there's any confusion, what we're talking about here, remember, is actually going to be inside of that planar part. When we're talking before about stereochemistry in terms of L and D amino acids. Here we're talking about like kind of like left and right-handed versions that are happening in the like non-superimposable mirror images that were happening based on what direction the side chain or R group stuck off of. And we said that most of that amino acids in our bodies are in this L form. This is going to be different. Well, in all those cases, however, we're still dealing with the trans amino acids usually, because that part that is referring cis or trans is referring to in this peptide bonds in this planar part that we normally can't rotate around. And so here this was where we're talking about the L versus D was which direction this R group stuck out in. When we're talking about this cis versus trans, here we're talking about the backbone in the planar part in the peptide bond, and whether it's zigzaggy, which would be trans, where you have these on opposite sides of the carbon bond, or on the same side with the cis, which is kind of more like a staple shape. But this is happening in that amide bond, in that planar part that we said we don't normally rotate around, which is why we need enzyme help to do it. But this R group is going to be where you have that L versus D. So don't get confused about that. Some other structural coolness about proline. So in addition to like normal protein, we can also modify protein. So we can modify it with this hydroxylation. And so a hydroxyl group is going to be this OH or this alcohol group. And what can happen is that the proline can get hydroxylated at either the 3 position or the 4 position, which is more common. So you can see it, you have this hydroxyl group added here, or you have this hydroxyl group added here to give you 3 or 4 hydroxyproline. And this can get incorporated into a special protein structure that is formed in collagen where you get a triple helix. So before we were talking about like alpha helixes where you had like a single helix, like a single chain was forming a helix. Here we have 3 chains kind of curling up around one another. And this gives you this really sturdy protein that can serve as an important sturdifier of your skin and your bones and your tendons and all this great stuff. But in order to make this, you need to be able to make this hydroxyproline. And in order to make this hydroxyproline, well, this is what we call a post-translational modification. So translation is the process where all these amino acids get linked up. And then if you do things to the protein after those are linked up, we call this a post-translational modification. And in the case of collagen, this post-translational modification we're talking about is going to be a hydroxylation. So this type of modification isn't as common as ones that we talked about, like phosphorylation, but it still does happen, and it happens in this hydroxyproline, which is incorporated into this collagen. I should also mention that in this collagen, you have proline kind of powering up with this other weirdo, the glycines. Remember, these were the 2 that had the really weird backbone angles. And you often find them together because proline's so restricted, and it's got these weird angles, but then glycine is really small, so it can kind of make it so that you can get back to your regularly scheduled protein afterwards or beforehand. And so in collagen, you have a lot of glycine, and you have a lot of proline and hydroxyproline. And you have a lot of collagen in your body because you have a lot of skin in your body. And then this collagen is going to be largely proline and hydroxyproline. And to make it hydroxyproline, you need to use an enzyme called proline hydroxylase. And this needs vitamin C. So vitamin C, or ascorbic acid, is going to serve as a cofactor or as a kind of helper molecule that's going to allow this proline hydroxylase to do its job. And so if you don't have vitamin C, which is the case with scurvy, then you can't make this hydroxyproline. And if you can't make this hydroxyproline, then you can't make the strong collagen. And if you can't make the strong collagen, then your teeth fall out and stuff. So not good. Some final notes about the biochemistry of proline. So We find it here in our weirdos. It has a hydrocarbon side chain. And so basically it's just hydrogens and carbons. Now we've looked before, and when we said that we had when we had hydrogen and carbon, they share electrons pretty fairly. So they're going to be like non-polar typically. And then they're not going to, when you have things that are non-polar, often they typically don't want to hang out with water, or water doesn't want to hang out with them. So water is going to be really polar. It's got those separation of charges. And so it's only going to want to let in other things that have a partial charge or that have a full charge, because remember that core tenet, the opposite charges attract and like charges repel. If you don't have any charges, there's nothing to attract. And so when you have things that are nonpolar, such as pure hydrocarbons, they're typically hydrophobic. So water is going to avoid them. Water is going to kind of form bonds to other water molecules that exclude those molecules, those nonpolar molecules, kind of forcing those nonpolar molecules to clump together. And so when we looked at other amino acids that had pure hydrocarbon side chains, so when we looked at isoleucine and phenylalanine and leucine and valine, in particular, these are going to be very hydrophobic because they only have those nonpolar groups. But if you look at proline, it's not as bad. Part of it's because it's smaller, and part of it is just because of its properties. But in addition to being less hydrophobic, it's actually because of its unique nature, where it has the ability to form those really useful kinks and turns. You often do find it on the surface of a protein. So you can find it in a variety of places, but you're not going to be able to find it in the actual like helices or strands and sheets and stuff. I should also, another thing to not get confused about is that this side chain is going to be, although it's kind of like a ring, it's not aromatic. And so when we talked before, we talked about these aromatic amino acids, tryptophan, phenylalanine, and tyrosine. What these have is they have this special kind of ring. They have a ring that has the resonance stabilization or this electron delocalization. So we talked before about how we had electron delocalization in the peptide bond. Well, these amino acids also have that delocalization, so that sharing between multiple atoms, they have that in the rings, in their rings. That is not the case with proline. With proline, we just have an aliphatic ring. So it's non-aromatic. We call it things that are not, basically anything that's not aromatic, we call it aliphatic, these hydrocarbons. So this is going to be aliphatic and not aromatic. So it's not resonance stabilized like that, but it is a ring. We can make proline, our bodies can make it, so it's considered non-essential in the dietary sense. So non-essential doesn't mean that our body doesn't need it. Our bodies do need all of these, but we can just make some of these ourselves, and this is the case with proline. So remember, in the case of scurvy, their proline was involved, but the problem was that not that you couldn't make proline, the problem was that you couldn't modify the proline because you didn't have that vitamin C. But you could still make the proline, and the way that proline is made, it can be made from the amino acid glutamate by this pathway. So basically what happens is that there's like a step that's going to be enzyme help, that an enzyme is going to help out. It's going to remove this oxygen, but then you make this really, really awkward structure, and the protein isn't very happy, and it kind of like spontaneously ringifies. It spontaneously ringifies, it loses water. You get this pyrroline 5-carboxylate, and then that gets reduced. So you add a hydrogen and you reduce this double bond to a single bond. And so this would be an imine, but what we have with proline is we have an amine. So we have this where we just have these single bonds to the carbons. And yeah, so then you end up with proline. You can also like go the reverse way. And so you can make things from proline that can then enter the tricarboxylic acid cycle. And so this is the TCA, the Krebs cycle, or the citric acid cycle. What happens is that in the case of proline, you can convert it into this alpha ketoglutarate, and this can enter the TCA and be used to make glucose, so we call it glucogenic. Finally, what else can we say about proline? Well, just kind of its name. So we saw the word pyrrolidine come up before. So pyrrolidine is where proline gets its name from. So pyrrolidine Proline, you can kind of just say pyrrolidine really quickly. It sounds kind of like proline. And it was first isolated in 1900 by Richard Willstatter. And that is the biochemistry of proline. So remember, it is an amino acid, but it's a secondary amino acid. When you link it up into to form chains, it's going to not have any hydrogens on that nitrogen in the backbone. This is going to make it so it can't form hydrogen bonds. And if it can't form those hydrogen bonds, it can't form those alpha helices and those beta sheets, and those strands and sheets. And so therefore, you're not going to give those strong secondary structure. Well, just a quick note, so the strands are like the individual strands, and then the sheets are where you have the strands kind of like join up with one another. But anyway, you can't do that with proline, and you're also even more restricted in what movement, the movement around the backbone in proline. So you're restricted to certain angles, which makes it really helpful for forming kinks in proteins, but not very helpful if you don't want a kink. And so that is the basics of proline. You also need it in your collagen. You need a modified form that requires vitamin C in order to make. So you don't have vitamin C, well, you can't make that modified proline. You can't make that hydroxyproline, and you can't make that collagen. And if you don't have that collagen, then your teeth fall out and stuff, which is what happens with scurvy. So that's the basics of proline, and hope it helped.